Okay, so we're continuing on the synthesis of fats, and so this is part 2, which we'll be talking about not only how we build these different types of fats that are important in our bodies, but also what their structure as well as the function is of these different fats. So Tri-Glycerides are going to be our storage form of fats that have a glycerol backbone and then have 3 fats attached to each carbon. And so this is going to be what is found in mostly in adipose tissue. This is also what the liver is going to build and package up into very low density lipoprotein particles, as we've talked about. Now, we do have different types of fats that are important in the body. So in our lipid membrane, we're going to have different glycerol phospholipids. Again, those are going to have the glycerol backbone. And within those, we have the aminoglycerol phospholipids, which of course will have a nitrogen group within the phospholipid. We also have a group of sphingolipids and sterols. So for our glycerol phospholipid, we have phosphatidic acid, which is the simplest form. And phosphatidic acid is basically going to be the glycerol backbone, which is those 3 carbons, carbon 1, 2, and 3. Attached to that 3 carbon is going to be your phosphate. So this will have an H2, and then we'll have our phosphate. And then we'll have, of course, a fat attached to carbon 2 and another fat attached to carbon 1. And so phosphatidic acid is going to be very important. It is important not only it has a role for building other glycerol phospholipids, but it also is a second messenger, and so therefore has a role in cell signaling. And then phosphatidylglycerol is a precursor for cardiolipin. And we'll talk more about that in a little bit. Phosphatidyl Inositol, as all of you know, is going to form second messengers. These are going to be important second messengers. So phosphatidyl inositol becomes IP2, IP3, and they can be important for regulating cytoskeleton, ion channels. They are oftentimes a docking site for proteins that are important in the membrane to come to. We've already talked about phosphatidyl inositol many times, and so that one's very important. Cardiolipin is going to be important for regulating mitochondria proteins. And it also is going to be essential for organization of mitochondria itself. The cristae, for example, is going to be dependent on cardiolipin. Now, our aminoglycerol phospholipids, we've talked about some of these. We've talked about phosphatidylcholine. Phosphatidylcholine is going to be important for our brain. Brain is going to use this to make acetylcholine. And then also phosphatidylcholine is all going to be, of course, a major part of the membrane. It's going to be very important with pulmonary surfactant, and it's going to be found mostly on the outer leaflet. Because it's on the outer leaflet, it's going to be involved in membrane-mediated cell signaling. Importantly, this phosphatidylcholine can also be transported between cell membranes, and it has a phosphatidylcholine transfer protein that does this. Phosphatidylethylamine, we've also mentioned, it has a role in the assembly of proteins that go to the membrane. So this helps to assemble. So it's sort of like a chaperone. So we talked about heat shock proteins. They help to for the mitochondria proteins that were encoded in the DNA go to the mitochondria, and then heat shock proteins would help fold the proteins properly. Well, that's sort of the job of phosphatidylethanolamine as well. So think of it as similar to one of those heat shock proteins. So it acts as a chaperone. And then phosphatidylserine is a component of cell membranes as well, and it's going to have an important role in apoptosis. And this is going to be on the inner leaflet, except during apoptosis. And it's actually going to be flipped to outer. Oops, sorry about that, outer. And by moving it to the outer, the immune system has receptors. So it can eat cell. So phosphatidylserine is sort of an immune receptor, and it tells the immune system that this cell is dying, you need to eat it. And then we have plasmalogens. Plasmalogens are going to help protect cells from oxidative stress and from cancer. And this is important because we always have radical oxygens, lipid peroxidation always occurs in cells, and so our plasmalogens are going to have what's called a vinyl ether bond. So this contains a vinyl ether bond. And this vinyl ether bond is highly susceptible to oxidation. And so it protects the other fats. So it's going to protect the other polyunsaturated fatty acids. And one of the important ones that it protects is going to be our essential fatty acids, arachidonic acid, which is in our second position. And that's usually arachidonic acid. Okay, so those are our glycerol phospholipids, and again, they're going to be part of the membrane. We've talked about a lot of these already. They're going to have that glycerol backbone. They're going to have different groups attached to the different carbons, which we'll discuss as we talk about building them. But what's important to understand is they are important. A lot of them are secondary messengers. Some of them are going to be important for communications. Others are going to have an important regulatory with respect to proteins, folding proteins, making sure that the formation of mitochondria structure is intact, etc. So next up, let's talk about our sphingolipids. So our sphingolipids are a little bit different than our glycerol lipids. Our sphingolipids have, again, these 3 carbons, 1, 2, 3, so that doesn't change. They also have a nitrogen through an amide group. And so if you look at over here, we have sphingosine here. And sphingosine is pretty basic. It has those 3 carbons again with that amide group. And it has 2 OHs and 1 R group shown here. And so that's sort of a basic structure of our sphingolipids. And our sphingolipids are important. Sphingosine 1-phosphate as well as ceramide 1-phosphate. So ceramide 1-phosphate, sphingosine 1-phosphate. So that means on that one position, they will have a phosphate. So remember, here's our 1, 2, 3. So over here, they would have that phosphate on that one position. These are going to be important mediators that are going to regulate things like cell growth, survival, migration, trafficking. They have a role in inflammation, and they also have a role with cancer and the development of cancer. Ceramides are also involved in apoptosis. And the reason for that is because some of your sphingolipids will get degraded. And so their degradation is going to produce ceramide. And so when you have a lot of lipid degradation, sphingolipid degradation, you have an increase of ceramide, that indicates that there's damage in the cell, and that can lead to apoptosis. So sphingomyelin is going to have different head groups. You can think of them. We have a phosphocholene attached, or an ethylamine over here attached. And so sphingomyelin is going to be important in nerve insulation. So sphingomyelin is nerve insulation. It's also going to be on the outer leaflet, so it's going to have a role in signal transduction. It's going to, one of the things that it does do with respect to signal transduction is that it's going to lead to the production of diacylglycerol. And so as we know, diacylglycerol, or DAG, does have a role in signal transduction. And then degradation of sphingomyelin will produce ceramide. And then too much degradation of sphingomyelin will lead to too much ceramide, and again can be a signal for apoptosis. So then we have our cerebrosides, and these are going to have a role in the immune system. And then we have our gangliosides. Now, gangliosides, you can see here, they have a little bit more complicated additions of oligosaccharides. And so gangliosides have these long oligosaccharides that kind of dangle out of the cell. So they're on the external side, or they're exposed to the extracellular matrix. And so they can be cell surface markers. So you can tell the difference sometimes between different cells based on the gangliosides that they're expressing. They're also important for cell-cell communication because they're sticking out so much. So they have a role in cell-cell communication. Okay, and then we have our sterols, our cholesterols. And so you can see we have our sterol nucleus, and on position 3, so the first ring is numbered in this direction. And so you have your polar head group, your hydroxyl group, at that position 3. So this is considered an amphatic compound, both hydrophobic and hydrophilic. And so cholesterol likes to embed itself in between your fats in your membrane. And again, it helps with fluidity and also to help maintain the structure of the actual lipid membrane, as we learned from Biochemistry 1. Okay, so now the basic structure of a phospholipid. So here's a tag again, and our tag has the glycerol as the backbone, and our phospholipid does have a phosphate group at that position 3, and then attached to that phosphate group can be different groups. And so typically on position 2, you have an unsaturated fatty acid, and that is typically going to be arachidonic acid. And arachidonic acid is going to be important for many reasons, which we'll talk about in a future lecture, but it really makes eicosanoids, for example. And your eicosanoids, your thromboxans, your platelet, your leukotrienes, things like that are going to be made from arachidonic acid. And arachidonic acid is made from your essential fatty acids. So attached to this phosphate group, you can have an ethylamine attached, a choline attached, etc. The building block of phospholipids is going to be this phosphatidic acid, where you have a phosphate group on that 3 carbon. So here we have on carbon 1, we have a fatty acid. And then on carbon 2, arachidonic acid, and then on carbon 3, we have our phosphate group. Okay, So we have fats converted first to phosphatidic acid. Phosphatidic acid has several roles. It's a precursor again for biosynthesis, so it can make many different lipids. It also has to do with a curvature of the membrane, so it has charge groups, and so it has an important role for fusion of vesicles with the cell membrane, for example. And it also has a signaling role within the cell membrane. So now synthesis of phosphatidic acid is basically going to be, you have your glucose, and then you're going to eventually get dihydroxyacetone phosphate. And then dihydroxyacetone phosphate through glycerol 3-phosphate dehydrogenase can become glycerol 3-phosphate. There is also in the liver glycerol kinase, and so remember the liver will pick up glycerol when fats are released from your adipose tissue, and so glycerol will be picked up, and then the glycerol can be worked on by glycerol kinase and generate glycerol 3-phosphate. Now the second part is activation of your fats, and that is going to be from fatty acyl-CoA, and you have an acyl-CoA synthase, which is going to use ATP to activate your fats. And so here's your first fat group, that R1, and it gets activated. And then you have a transferase here that's going to attach that to the carbon 1 position of your glycerol 3-phosphate. Then you can activate your second fat. So that's R2, is the same acyl-CoA synthase. It activates that, uses ATP again. And you have another acyl transferase which attaches the R2 group to that second carbon. And of course, phosphate attached to that third carbon leads to phosphatidic acid. So then what happens to phosphatidic acid? Well, some things to point out before we move to that is that you have in the liver and adipose tissue the convergence of wanting to make glycerol 3-phosphate, because your adipose tissue needs to make glycerol 3-phosphate so it can build tags, and so they can store tags. Your liver needs to make glycerol 3-phosphate, could be for several reasons, but in this case they're making tags too. And so they want to form those tags in the fed state, and they want to release them into very low density lipoproteins. Your liver and adipose tissue are both going to be able to have a dephosphorylation step, and then to make the diacylglycerol. The liver is the only one that's going to have this glycerol kinase, so that's different between the two of them as well. Okay, so now what's the fate of your phosphatidic acid? So we have the phosphatidic acid, we have our R1 group, our R2 group, and we have this phosphate group. And so phosphatidic acid is going to be the precursors for both tags. Attaching to that third carbon would be another R group, or you can get glycerol phospholipids formed. So the mechanisms is going to be different for the different types of head groups you're going to attach. So in the very basic sense, you have a different head group. It could be a serine, choline, ethylamine, and it attaches to that phosphate, and so you get a glycerol phospholipid. Okay. Alternatively, you can have another fat attached, another fat diacylglycerol. And so you have another R3 that gets attached to that carbon, and then you get a tag formed. This is going to be the product, of course, what happens in the adipose tissue is you're going to form tags. In your liver, you can make different groups depending on the needs of the cells, and livers, of course, can make things and release them too. So you have liver and adipose tissue produced in the similar pathway, so nothing really is different. The difference is in how they get there. So again, liver can use glycerol kinase, whereas your both liver and adipose tissue, and your adipose tissue always has to go through the DHAP to glycerol 3-phosphate, and then after you get glycerol 3-phosphate, basically it's the same pathway all the way down to your triacylglycerol. In the liver, your triacylglycerol is going to be packaged into very low density lipoproteins. In your adipose tissue, it's going to go to storage. So your triacylglycerol is going to be packaged in the liver. So this is occurring in the liver. And you're going to get the synthesis of the very low density lipoproteins. Very low density lipoproteins are going to have in it your tags. So that's that TG. It's also going to have some proteins, especially your ApoB100. And then it's going to have different cholesterols, as well as cholesterol esters. And then it's going to have different proteins. The proteins are all going to be synthesized on your endoplasmic reticulum, of course. Same thing as always done with proteins. Goes to the Golgi complex, goes into a secretory vesicle, and then it gets incorporated into the very low density lipoprotein particles. So on the surface is also going to be phospholipids. And here it's showing just the different makeup. So you have your phospholipids, you have your cholesterol, you're going to have proteins in there, you have your different fats in there, and they all get packaged into this particle. So the majority of the protein on this particle is going to be the ApoB100. And it's almost like you have one big long ApoB100 molecule that winds around the surface, which is shown with this blue sort of squiggly line that I just made yellow. So it's this blue thing here. So that's the ApoB100. The very low density lipoprotein particles are more dense than chylomicrons because they have a lower percentage of tags. Your chylomicrons are somewhere up here. So your chylomicron level is somewhere up here. This should be level. That's just a note. So you have less tags in your very low density lipoprotein as compared to your chylomicron. Very low density lipoproteins are nascent, similar to chylomicrons. So they're going to need to interact with a high density lipoprotein the same way the chylomicrons did and get the proteins C2 and E from the high density lipoproteins. So basically, your very low density lipoproteins are released from the liver, and in the blood, it will interact with the high density lipoprotein particle, and it will transfer to your C2, as well as your ApoE proteins. So those both come from your high density lipoprotein, similar to your chylomicron. So now it becomes a fully matured, very low density lipoprotein particle. So now the fate of of your lipoprotein particles and your triacylglycerols are the same as what happened in your chylomicrons. So you have lipoprotein lipase, which will cleave the tags. And it will release fatty acids plus glycerol. Nothing is different. Your ApoC2 is what activates your lipoprotein lipase. So you have your ApoC2 here. That's on the surface. Remember that came from your high density lipoprotein. Nothing changed. Your muscle has a low Km with respect to lipoprotein lipase, so it likes to grab fatty acids. And then your products, again, you started with a very low density lipoprotein, and you get an intermediate density lipoprotein and a low density lipoprotein as the products of this interaction of taking the tags away from this. Lipoprotein lipase in muscle, again, has a low Km. Lipoprotein lipase in adipose tissue is highest during the meal. So the activity is highest during a meal. Okay? So now, when you have a meal, your adipose tissue size, or the stores in your adipose tissue increases. And that makes sense. You're eating, so you have plenty of energy. So a lot of whatever fats you eat will go to storage. Adipose cells are going to synthesize the lipoprotein lipase when you have an elevated insulin-glucagon ratio. So increase insulin. The adipose tissue is going to pick up fatty acids. So this is going to be, in this case, from a chylomicron. So we have our chylomicron here. And the chylomicron is going to get rid of the fatty acids. Your adipose tissue will pick that up. And then your glycerol, your glycerol here, there you go, is going to be released from your chylomicrons, and their glycerol will go to the liver. And remember, this is the fed state, so glycerol is going to go to liver and interact with glycerol kinase and then form tags in the liver. So the liver will use that glycerol to make tags. This is very different than the fasting stage, where the glycerol picked up by the liver is going to be a precursor for gluconeogenesis. In the fed state, glycerol is used to make fat. The chylomicron will have a remnant, and so the chylomicron remnant will go to the liver and will be degraded. Your adipose tissue is also going to have the ability to generate glycerol 3-phosphate, and it can do this by a process called Glycero Neogenesis. So gluconeogenesis occurs in cells that need to make glucose. Glycerol neogenesis occurs in adipose tissue, so it can make glycerol, backbone to make fats or tags. And so for glycerol neogenesis, you can think of it as a shorter version of gluconeogenesis. And remember, we're in adipose tissue. Adipose tissue does not have glucose 6-phosphatase, so it cannot release glucose. So the point of this is not to generate glucose, instead is to generate the glycerol backbone necessary to make fats. And so you're going to have basically pyruvate to dihydroxyacetone phosphate. Right? And so that's shown here. You have your pyruvate carboxylase, adipose tissue has that. Oxaloacetate to PEP carboxykinase, adipose tissue has that. Phosphophenols eventually gets to dihydroxyacetone phosphate. And then similar to liver, you have the glycerol 3-phosphate dehydrogenase, which will then generate the glycerol for the backbone so you can make a fat. So glucose, again, is not going to be synthesized. But both adipose tissue and the liver have pyruvate carboxylase, and both adipose tissue and the liver have phosphoenol pyruvate carboxylase. And it's the same carboxylase, needs GTP, the whole thing. Nothing is different. So keep that in mind. Both liver plus adipose. Okay, so now we have this Tri-Glycerol cycle, and it has to be regulated, of course. And so you can think of it as a go-between between adipose tissue and liver, right? And so what you know is that when your adipose tissue picks up fats, it's going to release glycerol, and glycerol is going to go to your liver, and glycerol is going to become glycerol 3-phosphate, and then you're going to make a tag, and you're going to keep sort of this cycle going. Now, on the other side, you have the fasting stage. And so your fatty acids are going to be released for fuels, right? And then you're going to also release glycerol in that case, and then the glycerol will go to glycerol 3-phosphate. Okay, so there has to be something tying all of these together. So the cycle between liver and adipose tissue is actually controlled by the activity of PEP carboxykinase. Okay, PEP carboxykinase is going to limit gluconeogenesis and glycerol neogenesis. Okay, so when you have glucocorticoid hormones such as cortisol, cortisol is going to help regulate PEP carboxykinase reciprocally in the liver and adipose tissue. So one. So in the liver, you're going to increase both gluconeogenesis and glycerol neogenesis. And in the adipose tissue, there is no gluconeogenesis, but you're going to decrease glycerol neogenesis. And the result is that you get an increase of blood glucose and an increase of blood fatty acids. Okay, so now when you have an individual with type 2 diabetics, this is potentially a target for controlling the release of fats and the use of sugars. So there are drugs, and it's a class of drugs. Sorry. And these drugs are going to bind to a receptor on adipose tissue, and it's called the proliferator activator receptor gamma. And binding is going to activate PEP carboxykinase. So this is going to increase the formation of your tags in adipose tissue. Which is what you want to do because there's too much sugar, and so you want your adipose tissue to pick up those to be able to make tags. And so you want your adipose tissue to pick up the fats so that you're going to force your other cells to use the sugar because there's too much sugar. And so basically you're going to increase production of tags, and this results in a decrease of fatty acids. And this results in muscle plus other tissues using glucose. Okay, so as we know, fasting, you're going to release fatty acids from your adipose tissue because your insulin levels are low, and so glucagon will trigger breaking down fats and fatty in adipose tissue. So low insulin, high glucagon, and this leads to cyclic AMP, which leads to pKa, etc., etc., etc. You then are going to activate hormone-sensitive lipase. And this is going to cleave fatty acids from those triacylglycerols. Fatty acids get released, and they're going to be bound to serum albumin. Nothing new. And then they're going to be picked up by muscle, liver, etc. Muscle, of course, will use it for energy. Liver is going to make ketone bodies and also make acetyl-CoA, and it's going to use it for energy. The liver will pick up, you also have glycerol, and then this goes to the liver for gluconeogenesis. So nothing different. Everything's the same. You have in your adipose tissue, you have your tag stores. You have your lipases that are going to break off different fats and glycerol, release them. And this is nothing unusual. This is everything that we've already known. So the regulation is a little bit different. So as your adipocyte is going to be stimulated by the changes in the hormone levels to release fatty acids, the fatty acids can actually get re-esterified. And this is in the adipose tissue. And so this is re-esterification is going to prevent your adipose tissue from releasing all the fatty acids. And this is thought to have a role in controlling overall release of fatty acids from adipose tissue. So when your adipose tissue is breaking down all of its fats, it's not releasing 100% of its fats. About 40% of it gets recycled and goes back to forming tags. And so, again, this is going to be what's controlling. This is another control mechanism. And this is spontaneous, and this happens all the time. So the key enzyme, again, going back to this, is going to be phosphoenolpyruvate carboxyl kinase, PEP carboxykinase. PEP carboxykinase is going to be induced following activation of protein kinase A. Protein kinase A is activated following cyclic glucagon via cyclic AMP. Oops. So again, we have our protein kinase A. It's not really showing that, but anyways, so we have our oxaloacetate for PEP carboxykinase, and then this is eventually going to go to glycerol 3-phosphate to DHAP, make glycerol 3-phosphate, and then you can make more tags. And PEP carboxykinase is going to be induced by protein kinase A. And remember, pKa, and this goes to G protein, and then this goes to eventually cyclic AMP, and then this is pKa, and then pKa will activate in the adipose tissue PEP carboxykinase. Okay, so our fatty acids are either going to come from our diet or we're going to make them. We, again, are going to discuss different glycerol phospholipids, which, as I mentioned before, are very important not only as part of your membrane, but also as signaling. There are some that are going to be important for different compounds, such as lung surfactant. And arachidonic acid is going to be usually in that position C2, and it is a precursor for some very important pro-inflammatory and anti-inflammatory compounds, or eicosanoids. Our sphingolipids are also going to be membrane components. They're going to be important in protecting nerves, myelin sheath, and they also form glycolipids. So this is just an image that shows different components of the cell membrane and different lipoproteins, lung surfactant, etc. And some of them are going to have a glycerol backbone. Others are going to have a serine backbone, sphingosine. And so we have our tag, our traditional tag with our glycerol backbone. We have our different glycerol phospholipids, and that head group again can be a choline, ethylamine, a serine, an inositol, glycerol, or it can be a cardiolipin. We have our ether glycerol lipids, which include our plasmalogen, platelet activating factor, and this contains an ether group, so it has glycerol ether group, and depending on the different head group, it's going to be the different factors. We have our sphingospholipids, such as sphingomyelin, so it has the sphingosine, which again is that ceramide backbone, and it has a phosphate with a head group. And we have our glycolipids, and so this contains different glycolipids, gangliosides, etc. And so these are going to have sphingosine and different carbohydrates attached. Okay, so now with respect to how these are all made, again, fatty acids are going to be the precursors for all of these. Again, all of these, whether you're a glycerol phospholipid or a sphingolipid, you are a major component of cell membranes. Glycerol phospholipids are also going to be part of the blood lipoproteins, bile, and lung surfactants. And they are also a source of polyunsaturated fatty acids, especially arachidonic acid, which is going to be a precursor and again, this is usually at the C2 position. And arachidonic acid is a precursor for things like prostaglandins, thromboxans, and leukotrienes. So of our and of course, thromboxans are going to be important for blood clotting, whereas your prostaglandins and leukotrienes are going to be important in your immune system. Ether lipids are going to be our plasmalogen and plus our platelet activating factor. And our sphingolipids are, again, going to be very important for myelin sheath. Okay, so now let's go through a few of how the synthesis is actually done. So our glycerol phospholipids are going to be formed similar to our triacylglycerols, where you have a glycerol 3-phosphate, 2 fatty acids, One is going to be to a phosphatidic acid. And then you have two different pathways to add on different head groups. So again, you have your phosphatidic acid, which was formed from glycerol 3-phosphates plus 2 fatty acids and a phosphate. And then to that phosphatidic acid, you can first lose the phosphate, become diacylglycerol at that 3 position, and then you have a head group that gets activated, and then that head group gets added. So in the case of a glycerol phospholipid. So for this type of pathway that involves a diacylglycerol without activation and the head group gets activated, it's phosphatidylcholine, ethylamine, and serine. Now the second pathway is where you're going to phosphatidic acid is going to be activated. So in this case, your diacylglycerol has the CDP instead of the head group. And then the unactivated head group comes in. And this is for Phosphatidyl Inositol, cardiolipin, and Phosphatidyl Glycerol. Now, our synthesis of our glycerol phospholipids are shown here. So again, we had our phosphatidic acid. We have our first one, which is our diacylglycerol. And so we leave with diacylglycerol. And then we can have an activated ethylamine or an activated choline. And this will become a phosphatidylethylamine or a phosphatidylcholine. Now, why I'm showing this is because you can convert between ethylamine and phosphatidylcholine without going through phosphatidic acid by using 3 of these SAMs. So SAM is going to be S-adenosyl methionine. So it is a methyl group donor. This is a single carbon donor. And so you need 3 of those. So you're going to add 3 carbons to your ethylamine to make choline. Okay, so something else can happen with ethylamine as well. So we have an interconversion not only between ethylamine to choline, you have an interconversion of ethylamine to serine. So your phosphatidylethylamine your ethylamine can be swapped out by a serine, and you get a phosphatidylserine. So these are interconversions. So your phosphatidic acid goes to phosphatidylethylamine. And then you can get, let me move my interconversion. Oops. You can get with 3 SAM, a choline, or you can get a swap, and then this will be a serine. Okay, phosphatidic acid. So all that basically means is that there are multiple ways to make choline, and then there is a precursor of ethylamine to make serine. So by that second mechanism in which your diacylglycerol gets activated, remember this is the second mechanism, you have activation diacylglycerol instead of activation of your head group. This could lead to phosphatidylglycerol, which is here, phosphatidylglycerol, or you can have inositol, and you can lead to phosphatidyl inositol, shown here. Now, these can be further modified. So once you have phosphatidylglycerol, that's a precursor for diphosphatidylglycerol, which is basically cardiolipin. At the same time, as we know, phosphatidyl inositol is in the inner leaflet of the membrane, cell membrane, and that can be cleaved to our phosphatidyl inositol bisphosphate. So PIP2, which is important for signal transduction. Now our ether glycerol lipids, again, are going to, well, an example is plasmalogen. These are going to be synthesized by the intermediate dihydroxyacetone phosphate. Okay, and that's shown here. We have our DHAP. And so a fatty acyl-CoA will react with carbon 1 of your DHAP, and this is going to form an ester. So here is your fatty acyl-CoA, and it's going to form this ester. Okay, and then you get the fatty acyl-CoA exchange for an alcohol, and then your ether linkage gets formed. So you get a reduction, here to an alcohol, and then your ether linkage. Then you have the keto group of carbon 2 of the DHAP reduced, and then you get it esterified to a fatty acid with addition of the head groups. So you get, in this case, ethylamine attached, and so you have your first group, and then you have your ethylamine second group attached here, and then you attach an ethylamine head group here. And then this is going to give you a glycerol lipid, ether glycerol lipid. Now, this is just an example of the importance of lung surfactants. So you have your dipalmitol phosphatidylcholine, which is a major component of lung surfactant. And then, so this is just showing an inflated alveolus. And then without lung surfactant, you get collapsing of the sac. And so you need more pressure to reinflate. And then when you have lung surfactant, it reduces tension of the lining of the surface, and it prevents collapse. And so less pressure is needed to reinflate the sac. And so this is just, again, an importance of one of these lipids and their function in the actual body. Now, one thing that's important is you have to have a mechanism to reach out, especially to that A2 position, because that's going to be arachidonic acid. And sometimes you need to tap into your lipid to use these fats. And so we have different phospholipases that can cleave these glycerol phospholipids, and they do this by attacking the phosphoglycerol molecule at different locations. So we have our phospholipase C, and that cleaves phosphatidylate inositol to diacylglycerol and IP3. We have our phospholipase D, shown here, and this is going to release phosphatidic acid and an alcohol. We have phospholipase A2, which is important because it cleaves off at that C2 position. Phospholipase A2 is also going to repair damage by free radicals by swapping out the damaged lipid for a new lipid. And then phospholipase A1, of course, is going to attack that position at that one carbon. So again, phospholipase A2 we're going to hear more about when we start talking about the metabolites of arachidonic acid. So now we're moving on to our sphingolipids. So again, sphingolipids are important for cell-to-cell communication. It also is part of our AB blood groups. It is often a receptor for viruses. And it has a backbone of a ceramide instead of a glycerol. And it has serine as its makeup. So here we have our serine, and we have our fat, palmitoyl-CoA, and then you're going to get reduction, and then eventually, so you have a portion from serine, portion from that carbon-16 palmitate. The reduction is going to give you dihydrosphingosine, and then you're going to have fatty acids added to this, and then eventually this is going to lead to the backbone of the ceramide. Okay, so synthesis of some sphingolipids. So they're based on, again, ceramide. And then you have the addition of the head groups, so similar to what we were doing with the previous glycerol phospholipids. And you also have sugars that use UDP sugars. So here we have our ceramide, shown here. And then we can add our phosphatidylcholine. And then this is going to lead to sphingomyelin. We can also start with ceramide and have either UDP glucose or UDP galactose. UDP galactose would lead to a galactocerebroside, and this cerebroside can then bind, add to a sulfate, and the sulfate is going to use to make compounds that necessarily won't be soluble more soluble. UDP glucose can form a glucocerebroside, and this can become a globoside. It can also go on to form the UDP sugars, including things like sialic acid. So all of these have to be degraded in the lysosomes. And they're associated with deficiencies, diseases. So if you have deficiencies in being able to degrade ceramides and sphingolipids, then you're going to get accumulation and have various forms of diseases. Okay. Okay, so this is a galactocerebroside. And this is going to react with a 3' phosphoadenosine. And this is going to form a molecule which is going to be PAPS, 3' phosphoadenosine 5' phosphosulfate, or PAPS. And PAPS is going to be a major sulfolipid of the brain. Okay, so just a little bit on other things that can happen with respect to our adipocytes. So adipocytes can also make their own hormones. There are two hormones known to be made by these cells. It's leptin and adiponectin. So leptin is going to bind to JAK receptors, and so this is going to bind through that JAK-STAT pathway of signaling. It's going to be released as your TAG levels increase. So when TAGs increase, leptin is released. It's going to bind to your receptors in your hypothalamus. And then your hypothalamus will release a neuropeptides. And this causes cessation in eating. Okay, so this whole story right here is in mice. Now, translate to humans. Oops. Unfortunately not. So in mice, if you give leptin, then it's going to cause weight loss. When you give leptin to obese patients, it doesn't have the same effect. However, if you do give leptin to leptin deficient patients, this does induce weight loss. It's just that you cannot use this to treat obesity. It's thought to be due to, again, similar to a type 2 diabetic where you'll have insulin insensitivity. It's thought to be due to an insensitivity of the leptin receptors due to overstimulations. Okay. Adiponectin is another hormone. This could be linked, or it's thought to be linked to insulin resistance. So this gets released from the adipose tissue as it decreases This gets released from adipose tissue as the size of the adipocytes get larger. So adiponectin is going to bind to two receptors. It has a receptor 1, and it has a receptor 2. Binding is going to activate the AMP kinase. And a peroxisome proliferator activator receptor alpha, and both are going to lead to an increase of fatty oxidation in muscle and liver. Okay, and so both of these will increase and so this adiponectin is released as size increases. Okay, so just keep in mind fatty acids are going to be synthesized in the liver. Of course, we take them in during our fed when we're eating. They're going to be synthesized from glucose, as we talked about in part A. The glucose is going to go to pyruvate in the mitochondria and then eventually get out into the cytosol. In the cytosol, citrate will form acetyl-CoA and malonyl-CoA. And then you'll have fatty acid synthesis where you get two carbons added from malonyl-CoA, release of carbon dioxide, and then you get a building up of typically that 16 carbon palmitate. It's going to cost 2 NADPH and 1 ATP per cycle. The fatty acids get packaged. VLDL particles goes to adipose tissue. Lipoprotein lipase degrades the tags, pick up free fatty acids, etc. From your fatty acids, you can also generate glycerol phospholipids. You can also generate sphingolipids. And these are going to be important not only in cell membrane, but many of them have importance for protection. They're going to form different compounds, surfactants, cardiolipin, and you're going to need them for cell-to-cell interactions, communications, etc. So lipids have a lot of roles in just daily general homeostasis.